Hello, my name is Johanna Perens, and I'm an image analyst at a Danish biotech company called Cubra. And today I would like to talk a little bit about our 3D imaging platform at Cubra and also give you an overview how we have automated our microscopy workflows and data management, and then do a small introduction also into how we develop AI models for quantitative signal analysis. So Cubra is a contract research organization, and additionally we also do internal drug discovery. And for many of these experiments, we use light sheet fluorescence microscopy. And our studies are quite big, reaching from 50 samples to 100 samples even. So usually these experiments start up with the in vivo phase, where a drug is administered to an animal. We extract the tissue and stain and clear the tissue before we then light sheet fluorescence microscopy is being then done. And then as a last stage, we do image analysis. We work with very diverse organs from brains to hearts, kidneys, eyes, pancreas, so many different things. And we also have very diverse endpoints depending on the organ and staining and disease. And as we work with so many samples, we have needed to automate our pipelines for imaging. So we have equipped our microscopes with robotic arms and then built an automated pipeline around it. This pipeline can roughly be divided into 4 stages. So you have setup, automated scanning, automated post-processing, and QC. So in the setup, every cuvette with the sample is labeled with a barcode, and this barcode is attached to sample info and scan settings, which are inserted into a database using an app. So this is the first part that needs to be done before it's possible to start scanning. For starting a scan job that involves all the samples in a study, so they are scanned in a row, one by one, we also use an internally built app. And when this app is started, then the robotic arm will grab the cuvette and scan a barcode before it inserts the cuvette with a sample into the microscope. And by knowing what the barcode is, we can retrieve scan settings from the database and apply them, start scanning. After the scan is finished, this sample is also added again into the database and gets a label that it has been scanned. When the scanning is finished, the data is immediately transferred from scanning computers to high-performance servers for doing post-processing. Our data sets are quite big. They reach from 300-400 gigabytes up to 1.5 terabyte, depending on the organ and also number of channels used. We also use quite a high resolution for doing most of our scans. And for starting up post-processing, in every scan folder, we have also a barcode, which is then linked to the scan. And based on that, we can actually retrieve again post-processing information from a database and then start up stitching and downsampling of these data. Finally, the metadata of raw but also post-processed images are inserted into a database again, and very raw unstaged files are removed. Post-processed data together with QC images are then transferred to the large storage capacity server, from which we read to do further analysis. And the QC images, we always produce them automatically. And we produce, for example, meta slices taken at different orientations. We have maximum projections or different channel overlays. And our scientists will then evaluate these images together with metadata. And they then select high quality samples for analysis and then send the low quality samples back to rescanning. And this loop will then go long as all samples in the study have been fully finished and the data is high quality. As you can imagine, building this pipeline has actually taken quite a while and has been quite complex to find the most optimal way of building it. And we have encountered quite a lot of challenges on the way that I would also like to share with you today. One of them is, of course, that the metadata needs to be accessible for doing, for example, image analysis or AI model development. So, of course, for publishing data. And for that, we use very well-structured databases, and we encourage everyone to do so. Of course, if you have disruptions in the pipeline, you will end up with image data pileup, which is a big problem. And one way to avoid this is in the beginning, if you would know more or less what your throughput is, you can then predetermine your server sizes and connection capacities. This is extremely important. And of course, you consider parallelized batch post-processing or compression strategies too. Disruptions usually happen if there are third-party updates to software and hardware. And we have learned that it's very, very important to have active in-house support and people who know the microscopes, people who know the software and who also know the third-party software can help out quite fast. As I mentioned, the data might be very big and it's very complex and time-consuming to analyze this. So it's not the most important thing to keep your highest resolution possible. It's actually more important to downsample your data to the lowest possible resolution where you can still extract your endpoints depending on the application. Last but not least, how do you monitor sample quality if you have high throughput scanning? Right now we do it manually, but we are moving towards this, that we start using AI models that can flag anomalies in samples, such as, for example, blurry samples, samples out of field of view, or bubbles, etc. So to say, simpler things that are very easy to catch. So these samples can automatically go to rescanning. Exactly. So now a few words about how we do model development for quantifying signals in these images. The problematic here is that we don't only have many samples in a study, but these are also 3D data sets, meaning that you have not variation only in samples, but you have inside the 3D volume of every sample also quite big signal variation. So how do you do a quick model development in a standardized manner? And for this, we have come up also with a quite nice pipeline, which starts off by preparing the data, meaning that you will chunkify these big 3D volumes into smaller 3D volumes and then extract the middle patches. Here you can see patches from a kidney, from different areas of the kidney, and you can see how different they might be. This means that the model needs to see all these patches to be able to find exactly what it is interested in. So what we do is that we do take these patches and then we use pre-trained AI models such as Clip and Clip to embed them, meaning that we compress the information in these images into vectors and we can plot them as point cloud, which you can see on the right side here. So essentially every patch here or every point here is a patch. And you can see that similar patches actually cluster together. What you can start doing now is exploring your data set by using color coding, for example, color coding based on quality metrics or metadata. So first we explore the data and then we select some of the patches for training. Usually we start up with 10 patches and we use clusterings to maximize the distribution of the coverage. So finding patches that are most different from each other in the whole data set. Then we perform training data annotation, for which we use machine learning assisted annotation tools, different pixel classifiers basically, and then we refine the annotations further on. And then we perform model training, for which you can use architecture of choice. For example, we use 3D, 2D units. And then after the model training, of course, you will validate the model by first predicting the training batches, but also different samples, and then find out if you're satisfied with your model. If you're not, it goes into second iteration where you explore your data more. You find out on which IDs it didn't work or which signal types it didn't work, and you can add these patches manually. And so in that sense, it's a very iterative pipeline. And few learnings from this kind of embedding framework. We have noticed that it's very efficient in exploring and extracting training data. And it's it has been, or we have been able to achieve a lot higher quality AI models when we do guided extraction of training data rather than random sampling. And this framework may be very useful for finding outliers in the data set or comparing also different data sets to see if the same model would work on them. Very important here is that very few but high quality annotations always beat many low quality annotations. This has been a very good learning for us. And we always try to minimize the number of people creating these annotations to just not introduce additional variation in the data. Very important point is that teamwork achieves always the most accurate and generalizable AI models. So it's very important that biologists and data scientists and microscopists all work together. And I would also like to mention about if in your institute you are training a lot of models, you do need somehow to structure them and keep track of their stage and on which data they have been trained on. And you can use databases for that, and also actually make standardized pipelines for both training and applying models. Yes, and I would like to thank you so much for listening. I'm happy to take your questions.